Okie dokie. Okay, so this is spinal fluid. This is chapter 10 in your urinalysis book. So what I decided was because you don't your exam for urinalysis, exam 4, is not next week, the following week, which is nice. So basically next week I can just post a recording for semen analysis. It's a short PowerPoint, so that's going to be recorded. And then on Friday, I'll give you the rest of the UA lectures, which is synovial and serous fluid, and also bowel fluids, but that's also very, very short. So I'm going to be recording one of those lectures as well. Okay, so let's do CSF analysis. Again, this is chapter 10 in your book. I really, really do like the chapters for urinalysis. I actually enjoy reading these chapters, and I like the pictures in the chapters. They're very helpful. And chapter 10 talks about cerebrospinal fluid, which is CSF. And the pictures here are really nice. So I would definitely encourage you guys to, after you're done with this lecture, just go to the textbook and, just go through it. It's not a very long chapter, but it's nice because it has a lot of pictures and they're really nice and clear. And that's what you're going to need for this particular part of urinalysis. Okay, so on your own, you can go through these objectives for this particular lecture. Okay, so you may already know this, but CSF is a major fluid in the body, in the human body, and it provides the physiologic system. It provides a physiologic system to supply nutrients to the nervous tissue. It also helps in removing metabolic waste and produces a mechanical barrier to cushion the brain and the spinal cord against trauma. So it's got a very, very important role in the human body, the CSF. Oh, well, give me 1 second. Kaya's coming in. Okay. So the brain and the spinal cord are lined by something called the meninges. And there are 3 main layers of these meninges: the dura mater, the outer layer, the arachnoid, which is the middle layer, and then the pia mater, which surfaces of the brain and the surfaces of the brain and the spinal cord. The cerebrospinal fluid, the CSF, is produced in the choroid plexuses of the 4 ventricles. Okay, so remember there are 4 ventricles. There is the choroid plexus lateral ventricle. There is the choroid plexus of 3rd ventricle. There is the choroid plexus of the 4th ventricle. Okay, and about 20 mL of CSF is produced per hour in an adult, and the volumes in the adult is about 90 to 150 milliliters, and neonates is a little less. It's about 10 to 60 milliliters of CSF that's produced in the total volume of it. Here's a picture. This picture is actually from your book that I like because it shows you the layers of the meninges. So in A, what you see is the layers of the meninges of the brain. And I'm sorry, on the left side, right over here. And on the right side, what you're seeing is the layers of the meninges in the spinal cord. Okay, so as I said, you have these 3 layers: the dura mater, the arachnoid, and then the pia mater as well. And the same thing in the spinal cord, you have the dura mater, the middle layer, which is the arachnoid layer, and then the pia mater layer. Okay, so a little bit about the physiology, the formation of CSF in the physiology. The choroid plexuses are capillary networks that form the CSF from plasma by mechanisms of selective filtration under hydrostatic pressure and active transport secretion. The chemical composition of the CSF does not resemble an ultrafiltrate of the plasma. In the choroid plexus, the endothelial cells have a very tight fitting junctures that prevent the passage of many different molecules. And maintaining the integrity of the blood-brain barrier is very essential to protect the brain from chemicals and different substances circulating in the blood that could harm the brain tissue. So that's the reason why there are the endothelial cells have a very tight fitting fit because it wants to have that blood-brain barrier to protect the brain from chemicals and substances from harming its tissue. Okay, so the blood-brain barrier obviously is very, very essential to the brain, and we already know that there are chemicals that or substances that can be harmful, and so the blood-brain barrier won't let those chemicals pass through. So disruptions of this blood-brain barrier occurs due to certain diseases, and some of those diseases include meningitis. Sometimes it happens in multiple sclerosis, and this allows leukocytes, proteins, and additional chemicals to enter the CSF. Remember that the composition of the CSF is it's different from the composition of the plasma. And again, in these conditions that disrupt the blood-brain barrier, like meningitis or multiple sclerosis, this blood-brain barrier is disrupted. Okay, so how do we collect specimens from the CSF? Well, obviously we know, and you guys probably already know this, but we have to collect CSF samples by doing something called the lumbar puncture. Have you guys ever seen a video of that? Because if you haven't, I can find a good one and probably post it for you in the course. What we do is we collect this fluid, the CSFs, by a lumbar puncture between the third and the fourth, or the fourth and the fifth lumbar vertebra. So, this is not a very, very complicated procedure, but it does, you do have to have certain precautions when you are doing this procedure, because there can be risk of bleeding, and also it depends on the patient. So it's not just about bleeding, but also maybe keeping in mind that you want to prevent infection, and also keeping in mind that you want to make sure that you don't damage any of the neural, the surrounding neural tissue. The volume of the CSF that is going to be removed is going to be based on the volume available in the patient. So it's going to differ between an adult and a neonate. And also it's going to differ by the opening pressure of the CSF that is basically measured when the needle first enters the subarachnoid space. So the opening pressure, and basically whether the adult or whether the person is an adult or a neonate, is going to make a difference as to how much we're going to be able to obtain from this particular patient. If a patient has elevated pressure, that requires the fluid to be withdrawn very slowly and with very, very careful monitoring to prevent collection of large volumes. Okay, so that's when the pressure is really, really, really high. So the specimens have to be collected in 3 sterile tubes. Okay. These tubes are labeled. They are labeled 1, 2, and 3 in the order in which they are withdrawn. Okay. Tube number 1 is used for chemical and serologic tests. Okay, and these first tube is used for chemistry and serology, because these tests are the least affected by blood or bacteria that gets inducted or introduced into these tubes, because of a spinal tap procedure. So it's kind of like hard to avoid that you may have a little bit of contamination in there. And so that first tube would always go to chemistry, because chemistry tests are the least affected by these, these bacteria and a little bit of cells that could go in there. The other tube is going to be tube number 2, and that is usually designated for microbiology testing. So that's going to go for the microbiology lab. And tube number 3 is typically used for cell counting because it is the least likely to contain cells that are introduced by the spinal tap procedure. Okay, there are times that we can collect a 4th tube, and that may be drawn for the microlab to better exclude skin contamination. But however, a lot of times the supernatant that is left over is frozen and saved for any additional tests that may be needed. So don't discard the excess fluid that you collect in a 4th tube. You should freeze it until there's no further use for this, okay? And here is a picture of your CSF specimen collection tubes. You have tube number 1 that is going to chemistry and serology, tube number 2 that typically goes to microbiology, and then you have tube number 3 that goes to hematology for cell counts. Okay, and then you have some supernatant that may be left over that you could freeze until you don't need this anymore. So when you deal, it's very important that you realize how difficult. It's not that it's very complicated, but it's very uncomfortable for a patient to do a lumbar puncture, basically. So this is kind of like the type of sample that you can't drop on the floor, you can't leave unattended, because it's going to be very hard, especially if it's a very ill patient. And because of the risks that are associated with collecting a CSF, for you to try to get another sample from the patient. So you have to treat it with lots of care, because once you get a CSF, that's it for you. Okay, this is where you need to do your testing. And it's not comfortable for the patient. And because of all of the complications that can occur during the specimen collection, an MLS or when you're working on these specimens, you have to be extra careful. Okay. Ideally, these tests would be performed as a stat basis, but if that's not possible, then we can try to store the samples. Okay, so and we have to follow the protocol for hematology. The hematology tubes can be refrigerated for up to 4 hours. The microbiology tubes can remain at room temperature, and the chemistry and serology tubes are to be frozen. Okay. All right, so what do we do when we analyze these? All right, the first thing we're going to do is that we're going to take a look at the appearance of your fluid, okay, your CSF. So visually, we're going to take a look at these. The initial appearance of normal CSF should be crystal clear, okay, crystal clear CSF, okay? If it is not, then that can be an indication of something that's going on within the CSF, okay? So this is going to be a big part of your report. When you are reporting CSF, appearance is going to be something that you must note in the the paper or in the LIS system. Okay, so you'll have the choice to either say, okay, this is a crystal clear appearance. Sometimes some CSF samples look cloudy or turbid. Sometimes they look milky. Sometimes they look xanthochromic, and sometimes they are bloody or hemolyzed. Okay, so xanthochromia means that the supernatant, when you spin it down, is pink, orange, or yellow. Okay, and that means that there has been like the yellow formation of it, that there has been some sort of red cell degradation products that are present. So there's been some degradations of red blood cells. Okay, if you see different colors, like for example, from pink to orange or pink to yellow, that can mean the presence of oxyhemoglobin or heavy hemoglobin, or conversion of oxyhemoglobin to unconjugated bilirubin. So looking at the color of it is going to be very, very, very important. Xanthochromia can also be caused by the presence of carotene, protein, or melanin in the CSF. Okay. Let me just go back real quick to mention something about a cloudy or turbid and milky specimen. When you see a cloudy, turbid, or milky specimen, that can be a result of increased concentration of proteins or lipids as well, but it can also indicate the presence of an infection, with the cloudiness being caused by the presence of white blood cells. Okay. Okay, and here in this picture, what you're seeing is the tubes of CSF. So you're looking at the appearance of them. So from left to right, from left to right is normal. Then you have xanthochromic, a little bit of the yellowish color. Hemolyzed sample, which looks bloody, right? And then a cloudy sample. So if you can see it right here, there's like a line in the back of it. And right here in the last sample that's cloudy, you can hardly see the black line, right? That's because it's turbid or cloudy. And again, that could be, you know, high lipids, it can be high proteins. That could also be an indication of an infection, all right? So that we would probably have to look at that a little bit further. All right, so again, xanthochromia is a term that we use to describe the CSF supernatant that is pink, orange, or yellow. Okay, but there are different factors that can cause xanthochromia, and the most common factor is the presence of red blood cell degradation products. So hemoglobin, basically, because hemoglobin is a degradation product of red blood cells. Okay. The color can vary, and the variety in color that you see in xanthochromia comes basically from like the length in time the blood has been, you know, getting degraded. Okay, so that can vary, you know, from pink, you know, to yellow, all the way to probably an orange color. Okay. Sometimes when you do see xanthochromia, it could also be the presence of bilirubin, which is a pigment, and that gives it that color. Or again, like I said before, carotene, protein, or melanin that gives it that kind of color. So one thing that we have to be careful with is that sometimes we get a traumatic tap. A traumatic collection tap means that basically there was, you know, injury in the collection process, and you start to see grossly bloody CSF, which can mean that there was there is a difference. When you start to see grossly bloody CSF, that means that it can be bleeding that is actually happening, like intracranial bleeding or intracranial hemorrhage. Or the other reason why you would see something bloody in the CSF is because there was a puncture of a blood vessel during a spinal procedure. Okay, so that's what a traumatic tap is the puncture of a blood vessel during a traumatic tap. Now it's very hard because, you know, one could think that, wow, you know, I'm collecting a CSF that's bloody. This can be, you know, intracranial bleeding that's happening to the patient. There is a way to determine that, and there's a way, because sometimes we don't know when we're injuring the blood vessels as we're going in. So we have to be able to tell the difference between them. Okay, usually there are 3 visual exams or visual examinations of these samples that will help us determine the difference, and to determine if the blood is a result of hemorrhage or the blood, you know, this issue is coming because it's a traumatic tap. We just injured the blood vessel. Okay, so there are 3 things. The first thing is the uneven blood distribution. So that's what we're gonna look at first. Okay, when there is blood that is coming from the cerebral hemorrhage, like intracranial bleeding, it would be distributed. It would actually be distributed evenly throughout the 3 different tubes. Okay, 1 second, guys. So when the blood is coming from cerebral bleeding, okay, that will be distributed. Oh, 1 second. I'm so sorry. I don't know why I'm getting this emergency phone call. Okay, sorry about that. It was the wrong number and they were calling me like 5 times. I'm like, what is going on here? Okay. So blood from, let me just start over. Blood that is coming from hemorrhage in the brain will be distributed through the 3 tubes evenly. Okay, so remember how we collect 3 tubes. So you'll see even amount of blood in those 3 tubes. So if the blood is actually coming from a traumatic tab, okay, that will leave the heaviest concentration of blood in tube number 1, okay, with the amounts slowly or gradually diminishing in the rest of the tubes. So less blood in tube number 2, and then less blood in tube number 3. Okay, so that is, you know, a good indication. Just kind of look at it and see how, you know, the distribution of the blood is like in the tubes, and then you'll see a little bit more. Okay, so other ways too is whether there is clot formation. If there is a clot that is present, that it means that plasma was introduced into the specimen, and that is an indication of a traumatic tap. Hemorrhaging does not have enough fibrinogen to form clots, so you will never see a clot if it was really true bleeding into the CSF. Okay, other causes of clot formation in non-bloody CSF is due to damages in the blood-brain barrier or tuberculosis or tubercular meningitis, okay, which produces a web-like pellicle after refrigeration. Okay, another indication is looking at the supernatant and whether the supernatant is xanthochromic. Okay, xanthochromia is not present in a recent traumatic tap. What it indicates is that there was an older hemorrhage, like an older bleeding situation. And you could actually do a D-dimer test for the hemorrhages because that indicates fibrin formation at the hemorrhagic site. So that can be a little bit helpful to determine the difference. So those are the 3 things that you can do to determine if this red, bloody CSF is really coming from true bleeding or it's coming from a traumatic tap, okay? You guys can stop me at any time if you have any questions, please. Okay, so the next thing that we're going to do on the CSF samples is going to be a cell count, okay? The cell count is a test that we perform routinely on CSF specimens. And we typically count the white blood cells and the total cell count as well. Typically the red blood cells are not present in the CSF. And so red blood cell counts are seldom done, usually you know, seen from the appearance of the specimen, but if we see them, we really don't count them. But we're really concerned with is the count of the white blood cells in the total, you know, cell count when we're looking at CSF. So again, if you see red cells visually, you know, it could be again from a traumatic tab, and then you have to be able to tell the difference. Okay, Red cells are determined though when there is a traumatic tap that has occurred. So you can do a red blood cell count when that happens, and you can actually perform a correction for leukocytes or proteins if that is desired, if that is the protocol in your lab. The red cell count can also be calculated by performing a total cell count and a white blood cell count, and you subtract the white blood cell count from the total cell count, and that gives you the red cell count, okay? Granulocytes, when we prepare a sample for CSF cell counting, what we want to do is we want to make sure that this is a stat specimen, because granulocytes tend to lyse within 1 hour. So when you get it, you must do your cell counts right away. If you're going to do it manually, you're going to have to do it right away. The normal white count in the CSF, 0 to 5 white counts per microliter of blood. Neonates, however, can have a little bit of a bigger amount of cells, a bigger number of cells per microliter of blood, and that is still normal for them. So up to 30 mononuclear cells would be normal. So the method that we use is well, before I move on, I just want to say something. A typical specimen can have up to 200 white blood cells or 400 red blood cells, and that may still be as you're looking at it visually, it still may appear clear. Okay, so even if your specimen is clear, you may still have cells in there. And if it's over 5, that's abnormal. So just because you don't visually see it, it doesn't really mean much. You still have to perform your red blood, your cell count, okay? Microscopically, we use a method called the Neubauer counting chamber method. And that's a routine process that we perform to count the cells in the cerebrospinal fluid, okay? It's hard to use electronic cell counters because there's a lot of background counts, and this method has always been like a manual method traditionally, and it's still being counted this way now. Even though it's not the easiest procedure, it's still kind of like the standard procedure for counting the cells in the CSF. Okay, but automation, obviously, when it is available, automation with anything will be more precise. You know, it provides standardization from lab to lab. You know, if it's a stat specimen, it'll definitely provide a faster turnaround time for results. There are certain automated analyzers that are CBC analyzers that can perform cell counts. But in a lot of labs, I must say that automation is not performed, that they still do these counts manually. And I think you guys will see it get performed manually when you do your rotation in hematology, because that's where all the cell counts are being done. CSF cell counts at least, but there are different instrumentation that you can use, the ADVIA, the Sysmex, even Beckman Coulter has the ability to perform cell counts on CSF and things like that. Okay, so what we're gonna do is we're going to count ourselves using this hemocytometer. Okay, so the hemocytometer has basically two chambers. So the hemocytometer it's like a little, I don't know if I showed it to you guys, but it has like it's like a little mount, a little chamber that you put on the microscope. And it has two sides to it, okay? And microscopically, you can see it when you look at it, but it's really, really tiny. So in one side, there is a counting chamber. So this is called a counting chamber. And on the other side is the same counting chamber. So typically we count two sides and we average the numbers. Okay, so this Neubauer counting chamber, this is the entire chamber is divided into large squares. So I would say 1, 2, 3, 4, 5, 6, 7, 8, 9 different squares. Okay, each one of these squares, the the measurement for each one is 1 square millimeters for each one, okay? And we don't always count the entire thing. It depends on how many cells we quickly look at, we kind of like estimate. If there are way too many cells to count, we don't typically count all of these squares, but we count, maybe the center square, or maybe just squares 1, 3, 7, and 9, and then we just multiply, by 4, just to get a better estimate of the number. But it all depends on how many cells we initially see. But this is the standard Neubauer chamber system that we use to count our cells. So we load our specimen, we wait for it to dry a little bit so our cells are not moving, and then we put these chambers under the microscope, and basically we just move the microscope from one space to the other like this, all right, and then we count all the cells that are present, all right. It's not easy. All right, and then we use a formula. This is the standard Neubauer calculation formula for blood cell counts that is applied to CSF cell counts to determine the number of cells per microliter of blood. So you take the number of cells counted times whatever dilution was added to your sample. Sometimes we do have to dilute the sample, but sometimes we don't need to dilute the sample. It depends on your collection, okay, on what you have. And then we divide that times the number of cells counted times the volume of 1 square. Okay, so keep in mind that we may do 4 squares, we may do 1 square, we may do all 9 squares. So we have to put that into consideration into this specific formula. Okay, and basically what we want to do is we want to be able to use this calculation to convert the number of cells counted in a specific amount of fluid to the number of cells that would be present in 1 microliter of fluid. Okay, and again, we can do these in diluted or undiluted numbers. If you're going to use a dilution, you have to be sure that you add the dilution into your calculation. Okay, here's an example of a formula: the number of cells counted times the dilution times 1 microliter over 1 times 0.1 times 10, which is the volume counted, and that should give you the number of cells per microliter of blood. Per microliter of blood. So you want to make sure that you release them per microliter of blood. Okay, so again, I did say that you can use it in both diluted and undiluted. There are many other calculations that are available as well. You are going to follow the protocol that we use in the laboratory to calculate. There are some deviations from lab to lab, but we all have to follow the overall protocol from CLSI to be able to report the number of cells per microliter. Okay, but the purpose of using any calculation is really to convert the number of cells counted in a specific amount of fluid to the number of cells that would be present in 1 microliter of fluid. Okay, so that's the goal of using the formula. Okay, so we can do total cell counts and the white blood cell counts. These are the 2 typical counts that we will do on CSFs. So you, again, depends on the kind of specimen that you get. If you get a clear specimen, you don't have to dilute it. So that can be counted all undiluted, being that there is no anything else that you see in the under the microscope. When you do require a dilution, you have to use a calibrated automatic pipette, and use the right dilution factor and make sure that you add the dilution to your formula so that you get the accurate amount at the end after you've calculated your sample. Okay, in terms of white blood cells, when you do your white blood cell count, you have to be able to lyse the red blood cells so that they don't get in the way. So lysis of red blood cells has to be obtained before you do the white blood cell count on either diluted or undiluted samples of CSF. So you can use a liquid called 3% glacial acetic acid to lyse the white blood cells. And then you can actually add methylene blue to the diluent fluids, and that stains the white blood cells. So that's really nice because it gives you a nice differentiation between neutrophils and mononuclear cells, should you need to be able to differentiate them. But usually when we do the chambers, the CSF counting chambers, we just count white blood cells. Like, we don't have to determine mononuclear or neutrophils or things like that typically, unless your lab requires it. Okay, so it's also very important that we use the essential quality controls for CSF counting and also for other body fluids, because we don't just count cells on CSF. We also count them in serous fluid and peritoneal fluid and things like that. So we have to make sure that we perform the appropriate QC, and we can tell what are white blood cells in how they are different than red cells, and make sure that our controls are in and that we perform our controls before we can do anything else. So putting in your controls or performing your controls is going to be very, very, very important before you perform your CSF counts. All right, so after we do our counts, what we do is something called the differential count. The differential count is it's a way to identify the type or types of cells present in the CSF. And that's actually a very, very valuable test to tell us what kind of cells we're seeing, okay? Now, when you're doing a differential count on a CSF, it's very difficult. It is not an easy thing to do. It's very different than performing a differential count on a regular blood. When you do it on regular blood, you'll learn it really quick and it's easy. But when you do it on CSF, the cells appear very differently, and it is not an easy procedure. Okay, so the differential count has to be performed on a stain smear. You will not do a differential count from the chamber because typically those are not stained. And also, you won't be able to tell the difference from one cell to another by looking at it in the chamber. Okay, so you have to stain them, put them under the microscope, and then you can do your differential count. Okay, so with the diff count, you will count 100 cells, you will classify them, and then you report them in terms of percentage. Okay. We do have to make sure that before we do a differential count, we include the sedimentation, filtration, and centrifugation or cytocentrifugation procedures to be able to concentrate our specimens. So before we stain it, we take a drop of the CSF, we put it in something called the cytocentrifuge that kind of like holds a slide in like a vertical it holds the slide in like a vertical pattern, in a vertical stance, and then you add your drop there, and then you press the button to start the cytocentrifugation, and it sticks the cells to the slide. Basically, it concentrates the cells to the slide. Once you do that, then it gets stained, and then you can start counting your cells. But you must concentrate the specimen before you can actually do your differential count. All right, so this is cytocentrifugation. It forces the cells into a slide in a monolayer, and the filter paper that is included absorbs the moisture. So you'll put 1 ml of CSF to 1 drop of 30% albumin, and albumin tends to increase the cell yield and decreases cellular distortions. It's helpful for the counts and for the ID, the identification of the cells. Positively charged slides are used to attract the cells, and you can use daily controls of 0.2 mL of saline and 2 drops of albumin albumin stained for bacterial contamination. So this is kind of like a control slide to make sure that there are no bacterial contamination. There's no bacteria present in your actual sample. Okay, so that's centrifugation. In the book, you see a nice picture of the cytocentrifuge slide and how you put the slide in and the drop of CSF plus albumin, and then how it goes into the centrifuge to actually stick, the cells to the slide by force. And again, the albumin will help to retain the integrity of the morphology of the cells, which you're going to need if you're going to be looking at the kind of cell, right? That's what a differential is. Okay, so here's some cellular components that you're going to be identifying. The cells found in normal CSF are primarily lymphocytes and monocytes, okay? So this is the ratio. Adults usually have predominance of lymphocytes to monocytes, so about a ratio of 70 to 30. The ratio is reversed for children, so that's something to keep in mind. Sometimes you'll see occasional neutrophils in normal CSF, but sometimes that doesn't really happen all the time because neutrophils start to give us an indication of there's something else going on. So what you're really expecting to see is lots of lymphocytes and then some monocytes, okay? This is in the CSF, about a 70 to 30 ratio. Okay, so pleocytosis, okay, is the presence of increased numbers of these normal cells, okay? So when you see normal cells, but you see them in increased numbers, that's pleocytosis. And it's increased amounts of normal cells. Pleocytosis of normal cells is valuable in determining the cause of meningitis. So when you start seeing increase in neutrophils, that can be some sort of bacterial meningitis. But if you start seeing an increase in lymphocytes, that could indicate viral, tubercular, fungal, or parasitic infections or parasitic meningitis. So when pleocytosis involves neutrophils, lymphocytes, or monocytes, we want to be able to do our differential count to provide some diagnostic information about the type of organism that is causing this particular infection of the meninges or meningitis, okay? When you say meningitis, that's an infection of the meninges. So again, typically when you see a high white blood cell count of which the majority of the cells is neutrophils, then that can be indicative of bacterial meningitis. And if you see a white blood cell count that has a high percentage of lymphocytes and monocytes, that can be, that are elevated, that can be some sort of viral, tubercular, fungal, or parasitic meningitis. So that's something that we need to investigate a little bit further. All right, this table right here pretty much it says the same thing. Is the prominent cells that are seen in the CSF, lymphocytes. Again, they're normal. You may see different stages, which are fine. But if you start to see pleocytosis, which is an increase in these numbers, then you may be looking at a viral, tubercular, fungal, or parasitic meningitis. Or sometimes you see that in multiple, in cases of multiple sclerosis, especially with lymphocytes, sometimes HIV infections and AIDS, degenerative disorders, or parasitic infections. If you start seeing neutrophils, then you know the increase means bacterial meningitis, and granules may be less prominent than in blood. So that's going to vary a little bit. You're not expecting to see the morphology to be exactly the same as you would see it in blood. Okay, and neutrophils can also be seen in the early stages of viral, tubercular, and fungal meningitis, but these cells tend to degenerate rapidly. All right, monocytes, can be normal, but if you start seeing them in increased numbers, then we're looking at the possibility of viral, tubercular, and fungal meningitis, or sometimes multiple sclerosis. They are typically found mixed with lymphocytes. You may also see, here I have more here, macrophages, eosinophils, blast cells, lymphoma cells, plasma cells, epidermal cells, cordial cells, spindle-shaped cells, or malignant cells. And they all have some sort of significance here. All right, so I'm actually gonna go through these cells. I'm gonna look through the, I'm gonna show you the morphology of these cells so you understand what these cells look like. So these are the neutrophil cells. And again, if you see them, sometimes in very low numbers, they could be okay, but typically they're an indication of bacterial meningitis. And oftentimes what you'll see is that you see the neutrophils with cytoplasmic vacuoles that result from the cytocentrifugation. So these are like little tiny holes in here. These are vacuoles, and sometimes it doesn't mean anything, but it was the force of the centrifuge, because again, we're trying to force these cells to stick to the slide, that is causing some sort of distortion, right? Even though we put the albumin in, we don't want that to happen, but it's going to happen regardless. But again, neutrophils are associated with bacterial meningitis, and sometimes they may have some phagocytized bacteria. These neutrophils are white blood cells, and their main function is to, they're phagocytic cells. So they are cells that come and try to get the bacteria when, when the bacteria is present. Sometimes that's the way you see it with phagocytized bacteria. Here, there's more neutrophils. In the picture on the left, you see neutrophils with intracellular bacteria, but it's very hard to see. But I don't know if you guys can see the little bluish structures here. So that's intracellular bacteria that's within the neutrophils. Okay, so this in a CSF would be indicative of a possible bacterial meningitis. The picture here on the right shows you neutrophils with intracellular and extracellular bacteria. So if you see it here, some of it is intracellular and some of the bacteria is seen outside of the cell, so extracellular bacteria. Stop me at any time if you have questions, okay, guys? Another type of cell that you may encounter, and when you're looking at these differential counts in the CSF, is nucleated red blood cells. Okay, so that happens when there is contamination with the bone marrow. All right, this is, remember, it's a spinal tap. Okay, sometimes there can be contamination with the bone marrow, which means that you're pulling cells from the bone marrow. And NRBCs are typically not in the CSF, but they are, an indication that there was some sort of contamination. So seen with bone marrow contamination from a tap in about 1% of specimens. There are times that neutrophils that have something called pyknotic nuclei may look very similar to nucleated red blood cells. But pyknotic nuclei, when neutrophils have pyknotic nuclei, that means that those are neutrophils that are getting degenerated. So they're degenerated red blood neutrophils. You may also see, along with that, some capillary structures and epithelial cells from traumatic taps. Okay. But the neutrophils with pyknotic nuclei indicate degenerating cells, and they resemble NRBCs, but pyknotic neutrophils usually have multiple nuclei. So that's really the only difference. Here in this picture, you are looking at let's see, you're looking at bone marrow contamination too on the left, because you will see that there is some immature granulocytes, immature red blood cells in granulocytes as well. This is not very typical of a CSF, so there could have been bone marrow contamination here. And here on the right, you see some capillary and tissue fragments from a traumatic tap. Some capillary and some tissue fragments in here. All right. So with lymphocytes and monos, lymphs and monos in high numbers can be found in viral, fungal, and tubercular meningitis. When you see something called reactive lymphocytes, that indicates a viral infection, okay? In patients that have multiple sclerosis, you'll see that there are 50 or fewer lymphocytes per microliter, both non-reactive and reactive. And you see an increased number of reactive lymphocytes in HIV and AIDS. This picture right here shows you an image of broad spectrum of lymphocytes and monocytes in viral meningitis. So there are lymphocytes that are reacting right over here. So when there is viral meningitis, there could be a mix of lymphocytes and monos, and that's common, okay? Reactive lymphocytes typically contain increased dark blue cytoplasms and clumped chromatin, and are frequently present during viral infections. And you would also see those with normal cells, okay? So that's reactive lymphocytes. Eosinophils, as you can see here in this picture, are seen in parasitic and fungal infections, and sometimes in medications and shunts into the central nervous system. This picture right here, though, has a little bit of centrifuge distortion. So you see some vacuoles in here. Typically, you wouldn't see these vacuoles in these eosinophils, and eosinophils are white blood cells as well, but they have a little bit of an orange tint to them because of their granulation. But an increase in these numbers, in numbers of eosinophils, would be seen in parasitic and fungal infections. Macrophages can also be seen. Macrophages are designated to remove debris from the CSF. And the presence of macrophages in the CSF can indicate hemorrhage as well, okay? So the purpose is to remove cellular and other debris. It may be seen after repeated taps as well. When there is hemorrhage, they enter the CSF within 2 hours to phagocytize red blood cells. Red cells are degraded to hematoidin crystals representing unconjugated bilirubin. So you can see that in here, that after hemorrhage, these cells will come in to try to engulf the red blood cells. And then you see some crystallization, some hematoidin crystallization that represents the unconjugated bilirubin, which comes from the hemolysis of the red blood cells. So you see that in these little dark stained portions of that. Okay. These macrophages right here are showing some engulfed erythrocytes. So it's called erythrophagocytosis. There's more pictures of macrophages right over here. So let's see. Here on the left, you see a picture of a macrophage with aggregated hemosiderin granules. Okay, so you can see that a little bit darker in here. And then here on the right, just making sure I have the right description for every single one of these. So on the right, what you're seeing is macrophages with hemosiderin and hematoidin granulation. Okay, so do you guys see this here? There's a little bit of a yellow color to it, right? So this is a crystallization, a crystal formation here. I know it's hard to see, it's not that big. All right, so in the CSF, we're also going to encounter some cells that are non-significant. Clinically, they're not non-significant. We may see clusters of spindle-shaped cells like the ones that you see in the bottom. You may see choroidal cells that show different types of, like a really weird distorted border and nuclear uniformity. Okay, so here each one of these has its nucleus. So sometimes that just happens after a diagnostic procedure. Choroidal cells are the cells of the of the epithelial lining of the choroid plexus. They're singular, or sometimes you see them in clumps, and they are uniform cells. Sometimes you see epididymal cells lining the ventricles and neural canals. They are less defined with less defined cell membranes and usually are seen in clump. And then here in the bottom, what you see are the spindle cells that line the arachnoid. And typically these spindle cells, you see them in clumps. So the procedures, these diagnostic procedures that, may kind of like show, lead to these, the presence of these cells in the CSF are like the, a ventricular tap or neurosurgery, things like that can affect the, what we're seeing in the CSF. Okay, now you may also encounter abnormal cells, okay? So you may encounter malignant cells that are of hematologic origin, okay? So this is when we get into like the blood cancers, like the leukemias and the lymphomas. So lymphoblasts, myeloblasts, and monoblasts in the CSF are seen frequently in serious complications of acute leukemias, okay? In these kinds of cells, the nucleoli is very prominent, and actually it's more prominent than what you would see in a blood smear. All right, lymphoma cells are seen in the CSF, and they indicate that dissemination from lymphoid tissue. That's what that means. They resemble very large and small lymphocytes. They usually appear in clusters of large, small or mixed cells based on the classification of the leukemia. So we really don't know until we find out the morphology of it and the kind of leukemia that the patient has. These cells in the CSF may show you a nuclei that sometimes is cleaved. There is prominent nucleoli that is present that you also have to pay attention to. So here, the cells that you're looking at right here are here on the left. There is a cluster of cells. These are lymphoblasts, and they come from a patient having acute lymphocytic leukemia. It's hard to tell. We're not going to really, I don't think I'm going to drill this into your heads right now. I am going to get into this more when we do hematology, okay? So you really need to learn them when we get to hematology. But these are blasts, lymphoblasts, and they come from a patient that has an acute lymphocytic leukemia. Here on the right, you're looking at monoblasts, and these three big ones are monoblasts, and these two small ones are lymphoblasts. Okay, so you'll notice, lymphocytes, okay? So what you'll notice that these monoblasts right here, very big cells, right? They're blast cells, so that means they're immature cells, and they're coming from a leukemia. And the chromatin structure, which is what's in the center, like this purple part of it, in the nucleus, is very smooth, okay? It's not clumped, it's very smooth. And then you see these little eyes right here. These are the nucleoli. That's what we refer to as the nucleoli. And even these cells in the bottom have them as well. These cells in the bottom have nucleoli. The cells in the bottom are myeloblasts, and this is from a patient that has an acute myelocytic leukemia. And again, big cells, they're blasts, very smooth chromatin, and they do have nucleoli. It's a little hard to see right now because the picture is small. All right, and these are more cells of malignant origin. As you can see, you can look at the abnormality. You know that this is coming from some sort of malignancy in the blood. These cells right here are lymphoma cells. Some of them have the nuclei that looks cleaved, and some of them are not. Okay, so those are typical lymphoma cells. Let's see here. You're also looking at lymphoma cells with nucleoli right in there, like the little eyes of the cell. And then lastly, these cells here to the right are Burkitt's lymphoma cells. So these are, again, big cells, abnormal looking, but the only difference is that they have true vacuoles. These vacuoles didn't happen because of the centrifugation. They happen because that's Burkitt's lymphoma, okay? That's the way that it looks. There are also some malignant cells that come from non-hematologic origins, like cancers, like certain cancers. So metastatic carcinoma cells of non-hematologic origin are primarily from malignancies in the lung, the breast, the renal system, and the gastrointestinal system as well. There are also some cells that we may encounter that come from the primary, they come from primary central nervous system tumors, and those include astrocytomas, retinoblastomas, and medulloblastomas as well. So these are a little bit of more difficult cells that you tend to encounter. So this picture right here shows you a set of medulloblastoma cells. They come in clusters. They have nuclear irregularities, and they usually come in like a rosette formation. It's like it forms like a kind of like a little flower because they come in clusters. So they resemble flowers or rosettes. Okay, so the morphology in CSF is something that we do need to perform as a part of the hematology department. It's a lot of times it's part of the hematology, but sometimes it's part of chemistry. But in our hospital, it is a part of the hematology department for sure, right? Yeah, so this is really morphology. I'm not gonna really ask a lot of you when it comes to morphology here, except for you just knowing what's normal and what's abnormal. And then we'll get into the morphology more when we get into hematology, our hematology courses. So other than doing the, you're doing the what is it called? You're doing the appearance, you're making sure that, if it's a bloody specimen, it's not true bleeding. You're also performing, the visual inspection of it, the microscopic cell counts. After that, you're doing your differential counts. You're actually identifying the cells. You can also perform different types of tests on CSF, and you do lots of chemistry and microbiology tests, obviously, right? As you already know, because we pick up 3 different tubes, right? So we know that the first tube goes to chemistry and serology, the second tube goes to micro, and then the third tube goes to hematology. So the chemistry tests, we range from many different types of tests. We do protein tests in chemistry. You check the CSF serum, the CSF serum albumin index. You can check for glucose, CSF lactate, glutamine. So there are different things that we can do in chemistry, and also different things that we can do in microbiology as well. So let's start with the chemistry test. Is everybody okay? Any questions? No questions, right? All right. Okay. So the chemistry test that we perform often is the protein determination in the CSF. Remember that CSF is formed by filtration of the plasma, and you would think, okay, well, it's formed by filtration. Do we find the same, low molecular weight chemicals in the CSF that are found in the plasma? Okay. But there is this filtration process, and it is actually a selective process, and the chemical composition is controlled by the blood-brain barrier. So the reference values for CSF chemicals are not the same as the plasma values. They're very different, in fact. Okay. Some abnormal values result from, alterations in the permeability of the BBB, the blood-brain barrier, or sometimes due to increased production or metabolism by neurocells in response to some sort of condition. Okay. So clinically, it is important that we determine the chemicals that are in the CSF. and only a few of them, like the proteins, the glucose, the lactase. It's not a lot of tests that we can perform on the CSF, but when we do perform them, they are significant and they mean something. Okay, so the chemical test that is mostly performed on the CSF is the protein determination. So normal CSF has a very small amount of protein. The reference values for total CSF protein usually are listed as 15 to 45 milligrams per deciliter, but they are somewhat method dependent. So they can vary a little bit because of the methodology that you're using. And also the higher values of proteins are found in infants and also in people that are over the age of 40. The value of proteins in CSF is reported in milligrams per deciliter. So that's something to keep in mind, and not in grams per deciliter. Usually grams per deciliter is the unit that we use when we report plasma protein concentrations. Okay, in CSF protein concentrations, we're reporting in milligrams per deciliter, and that's the thing that that they're different. So that's something that we need to keep in mind. All right, so generally the CSF has protein fractions that are similar to those found in the plasma, but the ratio of CSF proteins to serum proteins, okay, varies among the different fractions of proteins, okay, because there are different types of proteins, okay. So for example, just to give you an example, in the serum, right, albumin makes up most of the in the serum, albumin makes up most of the CSF protein. But in contrast to the serum, okay, prealbumin is the second most prevalent fraction in the CSF. Okay, the alpha globulins are the haptoglobins and the ceruloplasmins. Another protein is transferrin, and that's the major beta globulin that is present, okay? And it's seen in the CSF, but not in the serum. Okay, CSF gamma globulin is primarily an IgG with only a small amount of IgA. IgM, fibrinogen, and beta lipoprotein are not found in normal CSF. So there is a difference between these proteins between the serum and the CSF. Okay, so what happens? What is the clinical significance of having elevated protein values? So elevated proteins or elevating values of proteins are seen in diseases, you know, in pathological conditions. They mean that elevated values can mean that there was damage to the blood-brain barrier, IgG production, immunoglobulin production within the central nervous system. There is decreased clearance, degeneration of neural tissue. Decreased protein values can be an indication of fluid leakage from the CSF. Meningitis and hemorrhagic conditions that damage the blood-brain barrier are the most common causes of elevated CSF proteins. And there are many other disorders that can elevate the CSF proteins, and finding an abnormal result on a fluid that is clear with a low cell count, it's not unusual, okay? It's not unusual. So let me show you here in this particular table some of the conditions that may be related to elevated protein results in the CSF. So it may be seen in cases where there is meningitis, hemorrhage, tumors of the primary central nervous system, primary tumors, multiple sclerosis, Guillain-Barré syndrome, neurosyphilis, pyeloneuritis, myxoderma, Cushing's syndrome, connective tissue disease, pyeloneuritis, which I repeated twice, diabetes, uremia, and decreased values can be seen decreased protein values in the CSF can be a result of CSF leakage or trauma or a recent puncture. Okay, so we have to be able to tell the difference, and that's why, you know, it's necessary to look at these values. So what are some of the methodologies that we use to determine proteins? There are two techniques that are mostly, they're commonly used for measuring total CSF proteins. And they use the principle of turbidity production and dye binding abilities. Turbidity production and dye binding abilities. The turbidity method has been adapted to automated instrumentation in the form of nephelometry. And methods for measuring CSF protein are available for most of the automated chemistry analyzers. So you can actually, you know, do this determination of proteins by automation, and you can find them in most labs. Most of the automated chemistry analyzers will have a CSF protein panel. So what are the protein fractions? Okay, so basically, to diagnose certain neurological disorders, it is important to measure individual protein fractions, because each fraction will give us an indication that something is happening, and it's kind of like a differential diagnosis. We were able to see the different types of neurological issues by looking at each protein fraction. Okay, so certain neurological diseases are associated with abnormal CSF proteins, and we have to do these measurements of the individual protein fractions. So proteins that show up in the CSF as a result of damage of the integrity of the BBB. So if there has been damage in the blood-brain barrier and there is a resulting protein fraction, okay, that results in a CBC, you know, that can be an indication of something. Okay, so we're going to be able to determine different neurological disorders here. So for example, multiple sclerosis will show you, a patient with multiple sclerosis will show you the highest concentration of IgG. Okay, we now have to look at the proteins that appear in the CSF and be able to tell the difference between what's happening in the CSF and then what's happening in the plasma. Okay, so albumin. Albumin is in the highest concentration. People that have multiple sclerosis can have the highest concentration of IgGs. To assess if the IgGs that is present is increased because it is being produced in the central nervous system, or it is increased because it is a result of a defect in the blood-brain barrier, we have to compare the serum and CSF levels of albumin and the IgG. So there is like a whole formula, like a whole IgG and albumin index that we have to follow. Okay, so we'll use the CSF serum albumin index to investigate the blood-brain barrier integrity, and we'll use the CSF IgG index to measure the if the IgG is being synthesized within the CNS. And then we compare. The comparison of the CSF serum albumin index with the CSF serum IgG index will give us some information. And again, this can be it can be done in automated chemistry analyzers. We can do albumins, we can do CSF albumins, we can do serum albumins, and we can do IgG CSF, and we can do serum IgGs. All right, so this will be the calculation of the IgG index. It's actually a comparison of the CSF serum albumin index with the CSF serum IgG index. And this compensates for any IgG that is entering the CSF via the blood-brain barrier. It is performed by dividing the CSF serum IgG index by the CSF serum albumin index, as you can see here in this formula. So the CSF IgG over the serum IgG, over the CSF albumin, divided by the serum albumin. And the values, any values over 0.70 indicate the IgG production that is within the central nervous system, within the central nervous system. We can also do electrophoresis or immunophoretic techniques to identify different proteins and fractions of proteins. The purpose of performing CSF protein electrophoresis is really to detect what we call oligoclonal bands. These oligoclonal bands represent inflammation within the central nervous system. The bands are located in the gamma region of the protein electrophoresis, and that indicates immune globulin production. So when we do a CSF electrophoresis, to ensure that the oligoclonal bands are present as a result of neurological inflammation, we have to run a serum electrophoresis at the same time. So we're kind of always comparing whatever's happening in the CSF to whatever's happening in the serum. Okay, when you have disorders such as leukemia, lymphomas, or viral infections, those can produce banding in the serum, which can appear in the CSF as a result of leakage of the blood-brain barrier. So these are things that we need to determine. We need to determine what is representing, systemic and neurological involvement, or any kind of infection or anything that is it's like a pathological, that indicates a pathological process. So basically the presence of two or more oligoclonal bands in the CSF that are not present in the serum can be a valuable tool in diagnosing multiple sclerosis. So multiple sclerosis, you see no bands in the serum, but you do see bands in the CSF. So do you see how it's helpful in determining certain conditions? Because what you don't see in one place, you may see in the other one. So that gives you an indication of something that could be going on. Particularly these bands that help us diagnose in the CSF, that help us diagnose multiple sclerosis, can be they can be accompanied by an increase in the IgG index. So it's nice when you're able to see the bands, plus you look at the IgG index. In this case, with multiple sclerosis, the IgG index would be increased. There are other neurological conditions like encephalitis, neurosyphilis, Guillain-Barré syndrome, other neoplastic disorders that also produce oligoclonal bands that may not be present in the serum. So we consider oligoclonal bands, we look at them, but we also combine whatever we find in the serum and in the CSF along with the clinical picture of the patient. So the history and what's going on with the patient really help to determine, ultimately what the probable condition is for the patient. Leukemias, people that have leukemias, lymphomas, viral infections, HIV will show bands in both the serum and in the CSF. Okay. All right. And let me see. I can move on to myelin basic protein. This is another major component of the myelin nerve sheath that surrounds the nerves, the axons of the nerves in the nervous system. And the presence of this protein, this MBP protein in the CSF, indicates destruction of the myelin sheath that protects the axons of the neurons. So this is called demyelation. Demyelation is destruction, okay, of this myelin sheath. Basically, the course of multiple sclerosis can be monitored by measuring the amount of MBP in the CSF, okay? It can also help us give a little bit of a valuable measure of the effectiveness of treatments. There's other causes of increased MBPs and sometimes CNS trauma, encephalopathies, Guillain-Barré syndrome, lupus, brain tumors will give you an increase in this protein as well. And you can use immunoassay procedures to identify or to measure this kind of proteins. We also measure in chemistry, we have the ability to measure the glucose. The glucose enters the CSF by selective transport across the blood-brain barrier, and this results in a reference value that is approximately 60 to 70% that of the plasma glucose. So the CSF glucose will always be at 60 to 70% and that of the plasma glucose. So you have a comparison, okay? It's never gonna be the same. If the plasma glucose is basically, 100, milligrams per deciliter, then the reference CSF glucose would be approximately 65. Okay, so that's the difference. So for if you want to do a plasma an evaluation of the CSF glucose, you can also do a blood glucose test to run for comparison. So the blood glucose actually should be drawn about 2 hours before the spinal time to allow time for the equilibration between the blood and the fluid. But you can use the same procedures for both. For CSF glucose, you use in blood glucose, you can actually use the same procedure that you would use for blood glucose. But you do have to test immediately because there can be glycolysis, especially in the CSF, and that will rapidly break down the glucose in the CSF. So you want to make sure that you test for glucose in a quick manner. Let's see. So what is the clinical significance of using, a glucose test in the CSF? So the diagnostic significance of glucose, it's really confined to finding values that are decreased relative to the plasma values. So what are my values as compared to the plasma values? Markedly decreased with increased neutrophils means bacterial meningitis. Okay, in tubercular meningitis, you'll see decreased glucose with increased lymphocytes, and in viral fungal meningitis, you'll see a normal glucose, but you'll see increased lymphocytes. So it gives us an indication of some sort of infection that could be happening, but we need to make sure that we determine what is the cause and what actually is happening here. So again, we compare whatever is happening in the CSF in terms of the glucose to what's happening in the plasma. Okay, so when you see elevated levels of CSF, that is always a result. It's always a result of plasma elevation values. Okay, it's never going to be the same number, but it's a result of the plasma elevation values. So when you see lower levels of CSF glucose, then that can be a diagnostic tool to determine some of the agents that are causing the meningitis. If you see, again, markedly decreased levels of glucose that comes with an increased white blood cell count, okay, and a large amount of neutrophils, then that indicates bacterial meningitis, okay? If the white blood cells are not neutrophils, but the white blood cells are lymphocytes, then you're thinking more tubercular meningitis, okay? But if you have normal values of glucose in the CSF with an increased number of lymphocytes, then that would favor viral meningitis, okay? So these are like little patterns that we need to follow to make sure that we get to the right diagnosis. Other tests that we can do are the lactate tests, the CSF lactate, and the CSF glutamine tests are additional tests that we can do in chemistry. The CSF lactate levels can be valuable in diagnosing in managing cases of meningitis as well. So in bacterial, tubercular, and fungal meningitis, the CSF lactate levels are greater than 25 milligrams per deciliter. And any levels that are greater than 35 milligrams per deciliter are seen frequently with bacterial meningitis, okay? And whereas viral meningitis, the lactate levels are lower than 25, okay? Just one thing to know is that CSF lactate levels remain elevated during the initial treatment, but they do tend to fall rapidly when treatment is successful. So it is a sensitive method for evaluating the effectiveness of an antibiotic therapy, looking at the lactate, the CSF lactate levels. All right. Also, hypoxia, okay, tissue destruction within the central nervous system due to oxygen deprivation or hypoxia can increase the levels of lactic acid in the CSF. Okay, so lactic acid is not, or lactate is not just limited to determining, the causative agents for meningitis, but it could also show that there are some sort of conditions that leads to decrease in oxygen blood flow into the tissues. Okay, so it's a good method to determine or to monitor severe head injuries. And glutamine, CSF glutamine, is another chemistry test that we can do. Glutamine is produced from ammonia and alpha ketoglutarate by the brain cells. This helps to remove the toxic metabolic waste product ammonia from the central nervous system. Ammonia is very toxic for the brain. The normal concentrations of glutamine in the CSF are between 8 to 18 milligrams per deciliter, and elevated levels are associated with liver disorders that result in increased blood and CSF ammonia levels. Excess ammonia in the central nervous system also increases glutamine synthesis. So determining the CSF glutamine also gives you gives you an indication of the presence of ammonia in the CSF. So you can also see elevated levels of glutamine in children with Reye's syndrome, in disturbance of consciousness when glutamine levels are more than 35 milligrams per deciliter. Okay, and this is a good test. It's actually more reliable than doing a direct CSF ammonia test, and it's you can perform it with automation. Okay, so lastly, we're going to talk about the microbiology tests that we do, and these are the last set of tests. Sorry, excuse me. Oh, okay. So in microbiology, the reason why microbiology gets these tubes of CSF is to really identify the causative agent of meningitis, because meningitis could be a very, very dangerous condition. It could be fatal. Okay, so bacterial meningitis, most likely could be very fatal if it's not caught in time, and it can be very dangerous for the patient. So we do some microbiology tests on this tube for CSF to identify, different types of organisms. Maybe we can recover the organisms into the appropriate culture medium. We can grow it in micro. Sometimes it takes about 24 hours, especially in cases of bacterial meningitis. Sometimes it takes about, 6 weeks, like tubercular meningitis, that takes a little longer. So the culture, is done, it's more like a confirmatory process rather than a screening process for these things. But micro does have several other methods that are available that can give us some information about a preliminary diagnosis, especially when we're really thinking that this is going to be a meningitis. So you can do a gram stain, you can do an acid fasting, India ink preparation, you can do immunological assays like latex agglutination, coagu agglutination, immunoassays, and molecular diagnostics with nucleic acid amplification tests and PCR that can help you determine these kinds of agents quickly, instead of waiting for these cultures to grow. Because again, sometimes especially these tubercular meningitis grows in like a long time, and it's, it can be very, very dangerous for the patient. Okay. So these are some of the organisms that we are concerned with. We are concerned with Streptococcus pneumoniae, which is a gram-positive cocci, Haemophilus influenza, Escherichia coli, Neisseria meningitis, the gram-positive cocci Streptococcus agalactae, and the gram-positive Rhizosteria monocytogenes can be encountered in newborn babies. Okay. Any questions? Okay, so cultures can be plated on chocolate agar when cultures are done. TB's meals are very important because of the growth time delay. Again, latest agglutination tests are available for these particular bacteria and organisms, and grain stains are best for detection. So every time we do something in micro with a CSF, we also want to compare it with the chemistry results and the hematologic results, because everything will give us a nice clear picture of what's going on. We can also do some testing for parasites, and the amoeba Naegleria fowleri is an opportunistic parasite. This one is found in ponds and lakes and pools. It enters the nasal passages and migrates into the brain. It is a mobile amoeba. You can see that in wet preparations, and it has an elongated tapered posterior area, and that is one that can be found in the CSF. We can also do serologic testing. We do, obviously you can do PCR and molecular testing on the CSF. But obviously cultures are definitely more sensitive, but it just takes longer, but it depends on the type of bacteria that you're looking for. Okay, so with serologic testing, on the CSF, you really mostly do it to detect the presence of neurosyphilis at a third stage. Because of the use of penicillin, early on, that was used to treat the early stages of syphilis, there's really not a lot of cases of syphilis nowadays. So the number for the request for serological tests to look for syphilis nowadays on CSF is pretty low. But you may see that you may get a serologic order like that here and there. But it is rare nowadays. Okay, so the CDC, so although there are many different serologic tests that there are for syphilis, they're available when you test the blood. There is one procedure that is recommended by the CDC to detect or to diagnose neurosyphilis. Is the Venereal Disease Research Laboratory, the VDRL procedure. But it's not as sensitive as the fluorescent treponema antibody absorption test, the FTA-ABS, but it is recommended for the detection of neurosyphilis. There's another test called the RPR. This is a rapid plasma reagent test, and it is not recommended because it is less sensitive than the VDRL test. If you do end up using the FTA-ABS test, you just have to take care, caution during the procedure and prevent contamination with the blood, because that will it can give you a false positive. So that's it. So this is one of the serologic testing that you may encounter while looking for syphilis. All right, so let me stop recording here.